Bismillahirrahmanirrahim. Please, host, moderator, please mute all the participants. We start now. Okay, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Peace upon on you.
Waalaikumsalam warahmatullahi wabarakatuh.
Good afternoon for Indonesian time. Very good morning in Poland time and European time now. And good evening, our friend in Taiwan, Korea, and Japan. Ladies and gentlemen, my name is Oktias Muzagi Lutfi. It's precious chance for me to be your master ceremony on this very special occasion. The third webinar and the workshop series event of World Class Professor Program, collaboration program between Universitas of Brawijaya, Indonesia, and Universitas of Szczecin, Poland. Excellencies, participants, ladies and gentlemen, before we proceed the program today, we would like to express our sincere gratitude to Excellency Chairman of World Class Professor Program, Professor Yenny Risjani, Excellency Speaker from Szczecin University, Professor Andre and Marta and also Eva, Excellency Speaker from the Spain, Dr. Emilio Soler Onis, and Excellency Speaker from Indonesia, Dr. Uun Yanuhar. Excellency, distinguished guests, delegates, ladies and gentlemen from around the world, honorable distinguished guest speaker, participant, delegates, ladies and gentlemen, algae have been used for a century in environmental assessment of water bodies and are now used in countries around the world. Algae are critically part of aquatic ecosystem that power food webs and biogeochemically cycling. Algae are also a major source of problem that threaten many ecosystems, goods and services. When abundance of nuisance and toxic taxa are high, thus algae can be used to indicate ecosystem good and services, which complement how algae indicators are also used to assess level of contaminants and habitat alteration. Ladies and gentlemen, relationship between algae, assemblage, and stressor caused by human help diagnose stressor and establish target for protection and restoration. Many algae responses to stressor have thresholds that are particularly important for developing stakeholder consensus for stressor management targets. So that today's seminar and workshop topic is microalgae culture and human impact. On this special evening, we have several agenda as follows. First, opening. Second is main event, webinar and workshop. 3 is question and answer session, and 4 is closing. Excellencies, participants, ladies and gentlemen, we are now going to the first agenda of opening. Let's open this webinar and workshop of microalgae culture and human impact with the word of Basmalah. Bismillahirrahmanirrahim. Excellencies, participants, ladies and gentlemen, we are now going to the main event of workshop in the webinar. So the first speaker today will be delivered by Professor Yenny Risjani, with the title of her presentation is Porphyridium growth and the benefit as immunonodulator. Prof. Yenny, Dr. Ph.D. Ph.D. from Nantes University from pronunciation in 1995, and she expertise on the marine biology, ecology, ecotoxicology, algae bioactivity, and biodiversity. Now he is a professor at the Faculty of Fisheries and Marine Science, Universitas of Brawijaya, and also founder of Ecotox Research Center. And related of her research in last 5 years, so many research you can see on this presentation today, and did not I will not read all, and you can read by yourself. And please, Professor Yenny, 20 minutes is yours.
Thank you very much. Thank you very much, Pak Okti. Good morning from our for our friends in Poland, and Assalamualaikum for our colleagues from Indonesia. Can you see our PowerPoint presentation or not?
Yes, clear, Professor. It's very clear.
Okay, thank you. I will show you. This is the slideshow. From current slide. Yes. Today I will share about Porphyridium growth and the benefit as immunomodulator for shrimp or shrimp faname. This is from our project funded by the DICTI, and we will focus in our talk here fibriosis. So we will talk about fibriosis and also again from the bioactive compound from Porphyridium crentum, and we will focus also to faname. So fibriosis is a disease that most often causes great losses for shrimp farmers, which can cause mass mortality in nearly 90% of captured shrimp in ponds. One alternative to overcome this problem is by giving antibacterial agents of the red microalgae Porphyridium. And faname, the shrimp is faname. It is the white shrimp, is the popular name. It is very easy to cultivate it, and the shrimp is highly productive, and it is able to live in higher range of salinity or euryhaline, and it is being adaptable to high culture density, as well as at a low protein feeding level. And based on phylogenetic position of Porphyridium from blast, we found the genus is the species is very close to Porphyridium purpureum. This is the strain with sample A primer forward and reverse. So this is the same result. And based on algae base, Porphyridium crentum is found by Natalie a long time ago. It is found on 1849. Yes, but the name has been changed to Porphyridium purpureum in 1960, around 1960, and the growth rate, the growth and culture of microalgae has been done by Nurul Mutmainah. This is former our master student from the thesis, her thesis, but it has been published in journal concerning growth rate and chemical composition of secondary metabolite extracellular polysaccharide in microalgae. So, microalgae growth was observed every day for 14 days of culture with the provision of iron, silicate, and vitamins, and continuous lighting in the laboratory. And this is the growth of Porphyridium, and we can see that the growth is has lag phase from 1 to 4th day and exponential phase from 4th day to 7 days and stationary phase from 7 day to 10 days of culture.
To the next speaker. Okay, I will invite the second presenter today will be delivered by Dr. Uun Yanuhar. With the title of her presentation is about the specification fragment protein from microalgae, C. Chlorella and Nannochloropsis Oculata, isolated from Java, C. Indonesia. Dr. Uun is a lecturer in the University of Brawijaya, and he got several achievement, like he ever been as the winner of the L'Oreal the young scientist from the L'Oreal. So
and woman in science.
Yeah.
Yes.
Yes, it's very nice and very experienced in many research. She has a 9 in H index and several publication in the international and local journal. So I give you like 20 minutes, Uun, to deliver your presentation.
Okay. I will share.
Okay.
Okay. You
Okay.
can see?
Yeah, we can see clearly.
Okay. Thank you for moderator, Pak Ogdias Musaki. Good evening for audiences in Indonesia and Asia, and then good morning for audiences in Polandia. Assalamualaikum warahmatullahi wabarakatuh. Honorable for Professor Yenny Risjani and committee for webinar on what class professor today. And I am Dr. Uun Yanuhar from Fisheries and Marine Science Faculty, Brawijaya University. I would like to present with title presenter, Specification Fragment Protein from Microalgae C. Chlorella vulgaris and Nannochloropsis Oculata, isolated from Java Sea, Indonesia. You can see the location of research. I have 3-part location. This is first part, and then second parts, and then 3 part in mapping this picture. The mic background. Microalgae is easy to obtain and easy to cultivate. When they cultivate intensively, microalgae can be harvested between 7 until 5 days. And then microalgae species produce carotenoid, antioxidant, fatty acid, enzyme, polymer, peptide, toxin, and sterol, and potential for the material for the antiviral and antifibrosis. You can see the collected my sample from the culture. And then about Chlorella vulgaris, found in all freshwater and seawater habitat, with main composition, protein, fat, carbohydrate, pigment, mineral, and vitamin, and then protein content 42 until 8%, and have pigment, beta-carotene, astaxanthin, sataxanthin, lutein, chlorophyll alpha, beta, thiopitin, alpha, and phyloxanthin, and have chlorophyll pigment can reach 1 until 2%. You can see specific performa cells from the Chlorella vulgaris from the culture. And then about Nannochloropsis Oculata, live in saltwater habitat, and for the source of dietary supplements, and have 1 chlorophyll A, and the main pigment is phyloxanthin, and have nutritional value protein, carbohydrate, fat, and vitamin C, and vitamin A, and calorie. I have collect sample from the outdoor culture, and then I'm separation with the filter, and then I'm collecting on the tube, and then I'm conduct to cell. This is a procedure. I'm isolating and characterization protein from cell microalgae with reset preparation and then microalgae culture and then isolation and identification of protein pigment fragment from Nannochloropsis oculata and Chlorella vulgaris, and then different concentration of PPF for the in vivo test to grouper or fish, and then the protein response of PPF again antigen like this Vibrio, and then viral nervous necrotic. The microalgae culture involves the media preparation, microalgae culture, fertilization, cell density calculation, harvesting, and isolation and identification. You can see the step for the culture, until the result, the outdoor culture. The sterilization of tool and media involved was all untilled rinsed with HCl solution and then soaking with 10 ppm chlorine solution for 1 day, and this with fresh water, like this picture. The laboratory scale culture one involve the isolate with agar media like this, and then culture of test tube and Erlenmeyer like this, and then culture in Erlenmeyer 5 liter until 10 liter. The semi-mass intermediate scale culture 2, prepare a fiber tube and aeration hose, and then fill a fiber tube with filtered seawater and add walnut fertilizer, and then microalgae starter is included as much as 20 until 30% and observe during culture, and I'm examine color change, bubble and foam, and then after 6 until 7 days of culture, and Oculata and Chlorella vulgaris can be harvested. This is the fertilization use is walnut with the following composition: ingredient aquades, ADTE, NaNO3, HEO3, NaH2PO4, MnCl, and FeCl3 with a dose and ingredients in this table. The harvesting can be done into two product, direct product, liquid precipitate or powder. I am collecting the pellet from the microalgae from outdoor culture, flakes from the microalgae and crush to mix microalgae powder. This is, and the sample screening of Nannochloropsis oculata. I want to screening from the sample Nannochloropsis oculata, and then process of centrifuges and oculata to screening result, and I am interpreting and analyzing data from the result. The protein isolation of Chlorella vulgaris and Nannochloropsis oculata with my modified method improved to patent number P00201709602 with specific isolation microalgae pigment protein fraction or PPF by ultra-high centrifugation at a speed of 250,000 RPM for 3 minutes 3 times. This is specific from the brick and swag protein from the Chlorella vulgaris and then oculata. For the next, the mixed sample, I for take 1 minute to collect protein, and then protein concentration, I am doing measurement with nanodrop test implant. Not forget, I am examine the cell with the same scan electron microscope from the Yanuhar 2040, and then for apply protein, a treatment for in vivo test, and apply in vivo test fish on pond. The protein isolation of Chlorella vulgaris and oculata. Step 1, I am collect from tube, and then I break with liquid nitrogen, and then I'm spiration with the ultra-high centrifugation for 250,000 RPM for 30 minutes, 3 times, and then I'm collecting supernatant, and I save for the deep pressure or liquid nitrogen. The step of SDS-PAGE involve the preparing sample by content like this, and then preparing SDS media gel electrophoresis with content like this, and then running SDS-PAGE, and then running sample, and then SDS-PAGE result media staining. I'm doing by Comesi brilliant blue staining and silver staining. This is a procedure for the collecting supernatan with high centrifugation, and then collecting the supernatan with yellow, and then so I'm conducting to electrophoresis, and then I'm interpreting data. This is a fragment pigment protein that's named pyridinine chlorophyll protein. That's a confirmed monomer with 33 kilodalton for Chlorella vulgaris, and then from Nannochloropsis Oculata 32 kilodalton. The report for the SDS-PAGE for protein Chlorella vulgaris and Oculata, I'm report to the journal Q2, and then the journal Aquaculture Indonesia, the accreditation national. This is specific for the fragment pigment protein that call it pyridinine chlorophyll protein monomer. This is from Chlorella vulgaris and then from the Oculata. The next step, second dimension electrophoresis was specifically conduct the protein. I would like to separate. This is on base the pH isoelectric point, where the protein separation conducted based isoelectric point in one dimension. Protein will analyze by dissolved in urea to conduct the hydrogen bond with used electrical area. And protein separates move and stop on specific pH when the protein load in neutral, Robbie load 1999. This is my result for the research. From the silver stain, PPF 35 kilodalton from Nannochloropsis Oculata, and the next step I'm separate with second dimensional protein. This is a two-dimensional distribution map of the 35 kilodalton PPF protein isolate of an Oculata, unpublished and on-progress manuscript journal. And then the next step, protein, I was to conducting RNA isolation of Nannochloropsis Oculata for complement DNA PPF conduct. Will be involve the cell separation and then breakdown of the cell wall and then the binding of RNA and finally washing and purification protein. The reverse transcriptation PCR or RT-PCR from the protein 35 kilodalton for PPF, Chlorella vulgaris, and Nannochloropsis Oculata. I have synthesis entry and amplification of complementary DNA with first primer for initial PCR and then second primer for nested PCR, and then I use RNA adapter primer because very sensitively for the PPF or pigment fragment protein. That amplification using RT-PCR was carried out is 35 pre-denaturation, denaturation, annealing, and elongation stage. This is RT-PCR. The primer use specific but double to RT-PCR. And then the next step, initiated primer, and then RNA adapter, and then continue to nested primer with temperature all T melting 5 degrees Celsius. This is a result of research transcription PCR or RT-PCR visualization from my sample from Nannochloropsis Oculata from C. Java with line 310 DNA. And then the protein confirmation of 35 kilodalton Chlorella vulgaris or cDNA PPF conduct 310 pp involve the genomic DNA extraction using CR plan and cDNA mini prep, and then PCR amplification using code VX neo to Yobo, and then PCR product purification with Simoclean, and bidirectional sequencing with step picture like this. And the sequence of phylogenetic of Chlorella vulgaris for C. East Java. You can see my sample with number 2836348 Chlorella P27F, and I'm report to NCBI. And then the report sequencing the protein fraction of PPF from Chlorella vulgaris with 16 single-strand RNA with initially guanine, cytosine, and then finally to IgGT. And I am report to the journal Q2 and then Elsevier Science Direct Proceeding Chemistry. Another result of the sequence of Chlorella vulgaris using primary RBCL, the isolate from Indonesia, C. Java, with 593 base pair, initially like this, guanine, cytosine, adenine, cytosine, cytosine, and then finally on the adenine, thymine, and I'm report to the Annual Basic Science International Conference 2019. The result of the analysis of the sequence Chlorella isolate local that have PPF 35 kilodalton and then 310 base pair. I am report the meeting algae in Berlin Aquaculture Europe 2019 with specific line 556 base pair with initially TTTT and then finally to GIGIIT. The specific response protein of 35 kilodalton BPF by Western blotting method. Initially, the sample was dyed with 0.2% containing 3% trichlorotic acid TCA, and then cut nitrosamine paper, and then with DH2 and block it with TBA, and rest again for 5 minutes using TBA, and the secondary antibody anti-VIS IG mice, and then second 2 hours and underwent 25 min risings processed with TBA, and then tablet chip dilute in H2O 10 milliliters, and then conducted an appearance of red color. You can see in the my result specific protein 35 kilodalton with examination of Western blotting like this. Patent now permit ID P002017109602, and then the next specification. Yes.
5 minutes to go.
Yes, until finish. Protein response of 30 kilodalton PPF immunofluorescence experiment with method cell tissue preparation and fixation and permeabilization and blocking and immunostaining and then counterstand, mount sample and observation. This is specification protein expression to 35 kilodalton with immunofluorescence and then measurement with peaks. And then this is, you can see, spiration response protein 35 kilodalton and measurement peaks. And then examination with immunofluocytometry, respond the 35 kilodalton on in vivo to Groper. You can see respond to CD4 and CD8. And then UV-VIS measurement to the concentration of protein. And then specific from the PCR of PPF Chlorella vulgaris, this is 3310 baseline. And then I'm collecting a protein, and then I'm collecting cDNA to cloning, gen, and then transformasi. And then protein pigment fragment PPF 35 kilodalton in vivo test. I'm use the VIS, and then the dye treatment and concentration, and then the specific test protein with hemagglutinin adhesion test, and I'm report to the Natural Pigment Journal. And thank you very much for team and committee, Professor Yenny, Professor John Luke, Professor Andre, Professor Junianta, Julie, Miriam, Badawi, Sulasti Arsad, Natalie, Roman, Ian Harifiler, and many other, and also research team, Nico, Nina, Professor Diana, Dr. Muhammad Musa, and Team Algae until 2015 until 2020. Thank you very much. I am return time to moderator. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Exactly 70 minutes. Thank you, Bu Un, for your nice talks today.
Yes, you're welcome.
So, ladies and gentlemen, now we continue to the next presenter. So the third presenter is will be delivered by two PhD candidates, Miss Marta Kriuda and Miss Eva Gorecka from University of Szczecin, Poland. So Marta and Eva is 30 minutes for you. So Marta, he took a BSc from Faculty of Geoscience, specialty on oceanography, and he took her MSc master on the University of Szczecin on Faculty of Biology and specialty on biological foundation of the forensic science. And oh, sorry, in PhD now is she is a PhD student in geography, and many scientific publication here. It's very small. I can read clearly. So next, Eva. Yeah, this is Eva Gorecka from University of Szczecin, Poland. She took a BSc from the Szczecin Faculty of Geoscience, specialty on oceanography, and he took her master science in 2015 from University of Szczecin, also Faculty of Geoscience, specialty on marine and coastal geography, and she expertise on the diatom isolation, culturing, molecular phylogeny, taxonomy, and monorapid diatom, practical application of diatom. She has also many scientific publications here. You can maybe ask to the Marta privately and Marta and Eva privately to talk about what they wrote on journal. So 30 minutes to yours, Marta and Eva.
Thank you, RTS.
Thank you.
Okay, please you can present by yourself.
Okay, can you see the presentation?
Yeah, okay. Yeah, it's okay.
So good day, everybody. Hello from Poland. My name is Ewa Górecka, and my colleague is Marta Krzywda.
Hello.
And we will be talking about culture and maintenance of the diatom culture. Firstly, we will talk how it all began for us for the Szczecin diatom culture collection, and then we will talk about how we sample, what kind of medium do we use, and then my friend will continue how we harvest, how we analyze, and in general, what can we do with our cultures. So it all began with a Maestro project funded by Polish National Science Center. The project leader was Andrzej Witkowski, Professor Andrzej Witkowski, and the main focus of the project was to analyze diatom assemblages in marine littoral zone of Atlantic Ocean, and also to investigate phylogenetic, biogeographical, and taxonomic implications. The project started in 2012 and ended in 2015, but we still maintain our culture, even though it began with the project, but now we also continue despite the project finished. The aims of our culture. So at the beginning, it was to study diatom biodiversity, as I said, and to apply multidisciplinary approach to determine the taxonomy and systematics of diatoms by means of traditional morphology approach and also molecular And we used, our standard is using 3 genes, SSU, nuclear encoded, and chloroplast encoded PSBC and RBCL. Also, our next aim was to study biogeography of marine littoral diatoms and in regional and global scale. Even though it was not original idea, with time we also somewhat shifted into finding the application for our cultures, and we tried to find application in industry. Advantages of diatom culturings are very broad. Of course, it's to examine their biology, morphology, and life strategies. So biology we can study by doing ecophysiological experiments. Morphology, so having diatom cultures allows us to have infinite fruits to see and to examine, and also life strategies. We can see how the diatom cultures live, how they produce, for example, stalks or other cellular components. Also, by doing molecular analysis, we can determine the phylogenetic relationships between selected taxa and their biogeography. And also we can check the dispersion in the global ocean. And of course, like I said before, to examine the potential application of diatoms in industry. So how do we proceed? So first of all, we go to the coast. We focus on littoral diatoms, on benthic, marine benthic diatoms. So what we really need is only a vial, and we just need to go to take a sample. So while we sample, we try to, if it's possible, we try to take all the metadata from the environment. We check the temperature, conductivity, we check pH, dissolved oxygen, and number one and the most important, we take the GPS position. We sample all sorts of substrates to like take all the diatoms from the environment, as many as possible. So we for water samples we use plankton net. We take also pieces of sediment, sand, small rocks, fragments of macroalgae as well. For epiphytic diatoms, also scrapes from the rock pools. So if the rock is too big to take it home with us to the lab, we just scrape, usually with a one-use toothbrush. We also take biofilms or foams, and also other, for example, turtle carapace, pieces of turtle carapace, which doesn't hurt the turtles, but it enables us to study epizoic diatoms on turtles' carapace. So while we take the samples, when we are in the field, we try to keep them in natural light, and if it's possible, we enrich the samples with culture medium to allow diatoms to survive. We use standard F2 medium with normal nutrients, with silica, trace metal, and vitamins. So when we take the samples to the lab, we use Petri dishes to transport diatoms from vials to Petri dishes, and we also enrich with F2 medium. For the inoculation and re-inoculation, we use inverted microscopes, and we use micro manipulation technique with just standard glass pipettes, and we elongate the tip of the pipette over the fire, so it's really, really small. We usually inoculate and isolate diatoms using 200 magnification with inverted light microscope. So basically, this is how the samples, environmental samples, look like when we take them from the environment, from the field, and we try to isolate the single cells to make monoclonal cultures. So only one clonal culture. So for now, I thank you, and my friend Marta will continue now.
Culture collection, we are placed into the grow chamber. Grow chamber allow us to control the environmental conditions. We can control humidity, temperature, and light. Our culture collection we store at 80 to 20 degree or 60 to 80 degree under a 12-hour light, 12 hours dark cycle, illuminated by 50 micromole of white light and humidity 50%. We also keep the culture in the 2050 milliliters flasks in the lab conditions, and also under the LED lamp in new culture room in basement. When monoclonal culture is established, material is splitted for molecular analysis. We keep pellets in the 1.5 milliliter Eppendorf tubes, and we store in freezer at minus 21 degree. I will talk briefly about molecular data. First step of molecular analysis is DNA extraction. We use for it Chelex 100 resin or genomic DNA nucleosplit plant to kit. When we do polymerase chain reaction, PCR, which is a method widely used to rapidly make millions to billions of copies of a specific DNA sample. PCR products we visualize in 1% agarose gel. PCR products we send to DNA sequencing laboratory EBBPAS in Warsaw, in Poland, for Sanger sequencing with use of big dye terminator chemistry and sequencer. And in the end, we built the phylogenetic tree.
Thank you very much.
Thank you.
Okay, thank you, Marta and Eva. It's nice talk. And I still remember last 2 weeks, we have question about the bioreactor from the Agency for the Assessment and Application for Technology from Indonesia. Maybe if she came today, she can talking to you and ask to you about the whole process of the bioreactor. So we continue to the next presenter. So it's welcome, Prof. Andrzej. So we continue to the next presenter. The presenter it will be delivered by Professor Andrzej Witkowski. On his presentation today, will deliver evaluation of human impact as revealed by the diatom assemblage in the various region sample. So Professor Andrzej got Ph.D. from the University of Warsaw, Faculty of Geology in Poland, and he now is a professor in oceanology and at the Institute of Marine Environmental Science of the University of Szczecin. His expertise is on the paleo aspect of marine diatom and is on species composition, biodiversity, taxonomy, phylogeny, and biogeography of marine littoral diatom worldwide. So, not what's the time? 30 minutes for you, Professor Andrzej.
Thank you. but yes, I am. How can I share my desktop?
Adrian, you can help.
Yes.
Click the green button.
Yes, I press the green button, and now
share a screen.
Sure.
Share screen. Yeah.
Okay. Oh, it wants my, okay, because it's my, let's hope. Is it?
No, not yet. This another, this a new email.
Oh, email, okay.
Nah, this is, yeah. This is.
Okay, okay. So, I'm so sorry for this misunderstanding. I've completely forgotten that we had a time change, and I was enjoying my breakfast when it turned out we shall start. So excuse my late show up.
Okay.
Yes, so we, as you see, we are the team which is working on the diatoms from around Indonesia. The diatoms, it is quite well known, are, well, let's say, are famous for bioindication. And whereas there is quite a lot of work done on bioindication in fresh waters, in Europe, each country is an obligation, must have the bioindication system, like each European Union countries and beyond the European Union. Also, Poland has this bioindication system. Our lab has been involved in establishing this bioindication system. In marine environment is maybe too much, little done is too much, but is still ongoing work on use of bioindication system to assess the water quality or the environmental health. Well, the diatoms for decades, the morphological approach has been used for bioindication of the environmental conditions. And now it's changing because more and more the barcoding, the markers, specifically SSU and RBCL, is used or is applied in water quality assessment, also in the marine environment. The advantage is of using the molecular data or metabarcoding is well, rather quick and good data, but the disadvantage or let's say the negative still the impact on the quality of data in molecular barcoding is impacted by the libraries, by the knowledge on molecular markers of particular taxa. So with each one strain isolated from the marine littoral, also from Indonesia, which Marta and Eva didn't mention, but you know we have quite a lot, about 50 strains. With depositing this data in the molecule, in the gene bank, we contribute to the success of the metabarcoding for the future. So let's hope that in a few years, both Poland and Indonesia will use metabarcoding as the standard method for the environmental, for the quality, water quality, for the environmental health assessment in the marine littoral. So but this is our ambitious plan. The recent situation is that we still use the data, molecular morphological data. You know, quite well known Sundaland and Wallacea, our target area. We started very modest sampling in south of Java. We moved later into the north of Java. We moved to West Kalimantan Barat, the coast of West and East Sulawesi, especially some islands of Wakatobi, and the area of Rinca, Komodo, and Flores. So you can imagine how much still is to sample if we take into account that Indonesia surface is over 1 million square kilometers. And this lovely target in our background, which I guess is Raja Ampat, is of course our target. So why it's so important to assess the environmental health of the littoral zone in Indonesia, because Indonesia is the fourth most populous country in the world. As you see, excuse me, yesterday Indonesia had 274,463,879 citizens. So the population is equivalent to over 3.5% of the total world population. The population density is in Indonesia is 151 person per square kilometer, but of course this is very variable from 1 or less than 1 to over 10,000 people in Jakarta per square kilometer. And as I said, it's not only 1 million, it's almost 2 million square kilometers. The 56% of the population is urban, and the median age is nearly 30. So as I mentioned, we have the areas which are 1 to 10 inhabitants, like in Kalimantan, Northeast, and the Papua, actually as a whole, or our famous Raja Ampat, but we also have Central and West Java with the population density over 10,000 inhabitants. So, and as you see, it's rather between 1,000 and 10,000 inhabitants in Indonesia. So the coastal area is very, the pressure on the coastal area is high, but this is actually, I would say, it's not easy to find a place where you can say in Indonesia the coastal area is strongly impacted by the human, by the inhabitants. This is why there are so many national parks. And when I told my colleague who is, let's say, a maniac in diving, about that we plan Raja Ampat trip for sampling, he was really excited. And the next, like Wakatobi, well, and Komodo, is there is no need to to make advertisement for these places. The coral triangle with some problems mostly related to ongoing climate change and ongoing human impact is still in, or is in transition, as is claimed by fish and coral and fish specialists. So now we can contribute to this pool of data with the information on diatoms, whereas still we will, I will be talking about very robust data. I can see the point where we will be able to determine the, or to formulate the values of the bioindications of diatom. So with this, I will say thank you and conclude that we have studied we have studied all sites we studied, except the north coast of Java. We have pristine waters. Even if we move eastward from Probolinggo, there is a change in the water environmental health as spoken by diatoms. So the site, the sampling site in the strait opposite Bali is already hosting a diatom assemblage typical for pristine marine waters. Terima kasih. Thank you. Dziękuję.
Thank you, Professor Andrzej. It's very nice talk and many positive news pieces, I think. Okay.
Yes, okay. That will be. I can say, well, of course you can, if you record this, you can prove it later. But I think 100 new species will be like very easy, and maybe 20 new genera.
Okay,
so
it's nice.
You can make a record, deposit this.
Yeah. Okay, thanks again, Professor Andrzej. And we now move to the last, our presenter is Dr. Emilio Soler Onis. He will present about the climate change effect in the Canary Island, HABs new from the distant island. Welcome, Prof. Soler So,
Yeah, let me know how I share with you my presentation. But I need to do the Professor Andrzej leave the screen.
Okay, okay.
Okay. Stop.
Yeah.
Okay. You can start now.
Did I make it correctly?
Yeah. That's okay.
Let me see. Compartir pantalla.
Hello, Emilio.
Yeah.
I will see.
30 minutes for you, Emilio.
Yeah.
Soler.
Can you see the?
Not yet.
Not yet. Oh. And now, well, we are a little bit far. Is now the presentation? Can you see the presentation?
Not yet, Emilio.
Yeah?
Not yet, not yet.
Oh, not yet.
So you can try again.
Okay, let me try again. Otherwise, could be very boring.
No, no.
Let me... Now? No. Not fin the presentation. A ver, compartir pantalla. Now?
Yeah, okay.
Yes, good.
And let me
drag your PPT.
I'm sorry for now?
Yeah.
Yes, that's good.
Okay.
Okay, so let me, yeah. Can you see the title?
Yes.
Yeah.
It's, okay. Okay, so first I would like to thank the organization of this workshop for the opportunity that you are giving me to be able to share the research that we are carrying out in the Middle East Atlantic Ocean, especially in the Canary Islands, in relation to the changes that are occurring in the marine ecosystems and their relationship with the climate change and potentially harmful algal blooms.
But Emilio, this is a presenter view.
Yeah.
So we can see your, you are not in our screen. So you can change.
I can try again.
Yeah.
Let me perhaps here. Now?
Still a presenter view.
Can you see the presentation or not?
Yeah, we can see it.
Okay. Okay, so many HAB are increasing in severity and frequency and biogeographical range. Causes are complex, but in some cases can be attributed to the climate change and human impacts, including eutrophication, habitat modification, and human-mediated introduction of alien species. But what is HEB? Harmful algal blooms is a result from noxious and or toxic algae that cause direct and indirect negative impacts to aquatic ecosystems, coastal resource, and human health. The term harmful algal bloom is normally a socio-economic but not a strictly scientific descriptor because it refers only to human perception of the harmfulness and the variety of negative events caused by algae, including some cyanobacteria and macroalgae, and present in nearly all aquatic environments, freshwater, brackish, and marine, and remember this, as natural occurring phenomena. Unfortunately, there is no plan and nor realistic possibility to eliminate this problem. Decades of research on monitoring have, however, improved to our understanding of HAB events, leading to better monitoring and predicting strategies. Let me, another one. The next one. It's not working. Okay, yeah. So far, there are around 5,000 species of microalgae identified in the marine environment, of which 300 of them can produce red tides. 80 of them have the capacity to produce powerful toxins that can affect the environment and, of course, the public health, especially in tropical areas. Let me, it's a little bit slow, yeah. So, no, sorry, yeah. There is news of its existence since biblical times, or more recently in the 17th century to present day. Don't forget that it's a natural phenomenon. Its expansion is linked to anthropogenic disturbances and environmental changes, such as global warming, ballast water transport, aquaculture, change in the coastline, eutrophication, and all these factors allow these organisms to appear in areas where they were previously unknown. And why are the HAB a global problem? Andrzej was talking about that before. Because about 50% of the world population lives less than 100 kilometers from the coast. This is a really, really serious problem because the future is going to be worse. Let me see, because yeah, there are 3 different types of HAB red tides. The first one is a species that can produce discoloration in the water, usually harmless, although under exceptional conditions in protected harbors from swell blooms. These blooms can appear and grow densely, causing indiscriminate damage to fish, especially to fish and invertebrates by oxygen depletion. I'm talking about Acasibo sanguinea, Goniaulas polygramma, Noctiluca, or the cyanobacterium Trichodesmium erythraeum. The second one is a group that the species belongs to, this group that produce very potent toxins that enter to the food chain to human, causing a variety of gastrointestinal and neurological syndrome. Probably this group is the most famous, unfortunately, in the human health. And the third group is related to the species that are not toxic for human, but they have a special structure. For instance, the Ketoceros, you can see the Keta, that they cause harm to the fish and invertebrates, especially in intensive aquaculture system. Ketoceros, Carenia, Carlodinium, a lot of Primnesophytes, and of course heterosigma, Akasiwo, and a big, etc. In the last decades, BHAB began to receive more attention since the center harmful benthic species, mainly of tropical origin, have been identified, and this is a problem, in temperate regions where they have become established. BHAB, or harmful benthic algal blooms, refer to the growth of benthic microalgae, which can pose a serious threat to other organisms, the environment, and the public health, because they produce potent toxins like ciguatoxins, myotoxins, palytoxins, ovatoxin, ocadic acid, and limbiatoxins. And if you can see here the map, look at the big distribution in the world, especially in Indo-Pacific area. Okay.
Okay. Okay. Thank you very much, Dr. Emilio Soler Onis. Very long talks and very nice talks. For me, is quite new. Okay. So next session is we are now going to the question and answer session. So everybody who want to talk to the presenters today, you can firstly introduce yourself, and you can put your name and department, and please make the simple as possible, short as possible question. So we have 15 minutes to question and answer. So in my chat box, I read from from Bu Retno, from Marine Science, Diponegoro University. Bu Retno, you can ask directly to Ibu Uun. Okay, no. So Bu Retno have question here to Dr. Uun. When you use freshwater chlorella, would you have the same result? What actually the difference between freshwater and marine chlorella? Uun is yours.
Okay, thank you, Pak Pias Musagi, for my time. My answer for Dr. Retno, the differences between freshwater and seawater chlorella from isolate Java is in the content of protein, which is on seawater is 32%, and from freshwater is 80 until to 22%. But there is nothing different morphology of chlorella in freshwater and seawater. And then based on our research, we can find PPF on chlorella from freshwater. And then specifically from the seawater. And seawater chlorella have terpenoid was found, but not in freshwater. And species from seawater can be cultivated by freshwater, but it needs adaptation process to the freshwater media and need longer time for the freshwater. Thank you, and I'm written time to Pak Pias. Thank you.
Okay, thank you, Bu Uun, for your answer. So now is time for you, all participants. I see in here Eris Sahabudin from Japan also.
Oh.
Yes, please, Eri Sahabudin, if you have any question, please.
Okay. Thank you so much for the time and for the invitation to join this seminar. First, here I have two questions. The first question belongs to Professor Andres. He mentioned that some report from of the atom from Indonesia, and also he mentioned about the atom from the cool region like Russia. My question is, what is the different characterization of between the atom in Indonesia and Russia? And also, as we know that now we are facing the global warming, which area is the most the global warming will be affect most, whether it's in the Indonesia, in the what is in the cool region or in the hot region. And my second question is belong to Dr. Uun Yanuhar, and as she mentioned that she used open pond system in harvesting the microalgae. My question is, when you use the open pond system, how you control the contamination, and then whether and you also use the aeration system. Did you also use the carbon dioxide or only use the air only? Okay, thank you.
Okay, thank you for your question. Question is directed to the Prof. Andre and Uun. Please, Prof. Andre, time is yours.
So thank you for the question. Well, it is in the following my presentation by Dr. Emilio Soler. It was evident that the temperate areas are victims of the increasing or the increase in the ocean water temperature, because the not only diatoms, especially the potentially toxic taxa migrate either with human help or with oceanic currents to the temperate areas, and it is I mentioned England and China. So I think the smallest diatoms, like I mentioned, Minidiscus trioculatus, they possibly migrate with the global conveyor belt, with the ocean currents. This is one of the mode of transport. The other one, I mean natural transport, is the wind system. The stratospheric winds are circulating the world, and they deposit somewhere what they are transporting. So it's long distance and short distance wind system. And of course, the human impact, the exchange of the ballast waters. I edited a book in 2016 on the transfer of viable diatoms in the Atlantic and Pacific coast of Canada. So there is a lot of diatoms which are viable, able to germinate and to spread in the new habitats. The interesting or the tricky point of diatoms is that they may produce the resting stages. That means they can survive a year or 2 years or even more in the ballast water. And once they are released into the light with the exchange of ballast water, they will germinate and they will be happy in, they can acclimatize to the new conditions. So that's the reason why we have, and we have checked this with the molecular markers, that the tropical area has a lot of cosmopolitan worldwide distribution species. We can't speculate now about the reason they, like the temperate water taxa, how did they get to Indonesia or to Australia or to any island. But even the most isolated islands now have their cosmopolitan species, and this is in part natural phenomenon, and this is in part caused by the humans. So that's what I can answer now.
It's clear, I think, Professor Andrzej, thank you. And now to Dr. Uun, please.
Yes, thank you for my question. From Iris Yabudin from Japan. Yes, I am harvesting and control for culture indoor and outdoor with sterilization, and then the wash all utensil, all the prepares for culture, and then every time I'm rinsed with HCl solution for laboratory culture and intermediate culture, and then I'm checked every time the contaminant like this bacteria, gram positive and negative, and then soaking with 10 ppm chlorine solution for 1 day, special for the intermediate culture and the outdoor culture. Yes, and then after outdoor culture, I will rinse with the fresh water. This is every time and respectively again. Thank you, and I'm return time to moderator.
Okay, thank you, Dr. Uun, for the clear explanation to Iris Yabudin. And now, to the next question came from Ibu Nurul. The question addressed to the Dr. Emilio. So the question is there any characteristic to identify the toxic microalgae and the useful one? So how can we difference the useful and bad microalgae by looking for the ecosystem condition? So
I'm sorry because I couldn't listen properly because it's
okay.
Maybe Mbak Nurul can directly give question.
Okay.
Sorry because our connection is not good.
Yeah. Yeah.
Chatting.
My connection is not really good, so I stopped the moderator, the text.
Okay.
You can use
but
chatting.
I'll try to. Oh yeah.
With chatting.
I'm so sorry.
Yes, by chat. Yes.
Okay. Dr. Emilio, you can check the chat box. There is any question for you from the Professor Yunianta?
Okay, let me.
Chat box, yeah.
Hello, Miguel. Hello. It's not far from the Sahara, so I want to ask you about the effect of Sahara sand, for example, silica content. Well, you mean the silica content?
Yeah.
Yeah? Well,
yeah.
Not really. The most important or the most interesting thing about the Sahara dust is comes from an area is very rich in phosphates.
Phosphate.
Yeah, and we were trying to know the composition of this Sahara dust, and of course there is a few cells of we can find a few cells of diatoms. They are flying hundreds of kilometers, but I think the silica is nothing if you compare with phosphate and of course iron. The iron is the most important factor to the growth, especially the for the bio assimilation. So silica from this in this area is not important, or is nothing. I'm sorry.
Okay,
But
thank you.
I have a question for Andrzej, for the Professor Witkowski.
Yes, okay, please.
Yeah, it's concerning about Luticola, because also we found here Luticola in the Canary Islands of 3 different species, but they are growing in caves, in volcanic tubes. And if you compare the temperature between volcanic tubes and outside, inside the volcanic tubes is around 10 degrees, and outside we used to have 29, 30 degrees. So our species are related with a circumboreal species. What about these species in the Pacific area or Indonesian area?
So thank you for your question, Emilio. It is, in fact, we also have the European junk in we studied, right now we are submitting paper on 3 new species from 1 sample from Easter Island, Pascua. Isola de Pascua, is it?
Isla de Pascua?
Yes. Which is the most remote island in the world, inhabited island. And we found them in the cave, the 3 new species and 1 tropical species, cosmopolitan. But in Indonesia, in Java, we found only these 2 species, which seems to be limited in distribution. But Sulawesi is like a paradise of Luticola, and the other area which is a paradise for Luticola is Antarctica. More than 40 species in Antarctica and subantarctic islands, and there is the cosmopolitan species and species new to science. Are your species named? They have names.
Yeah, well, I'm writing the article. They are new for species for the science. My name is Luticula parva because it's really small.
Very small.
Really, we are talking about 10 microns.
Micrometer.
Length.
Yeah.
And the other is Luticola. The name is Luticola. I'm sorry because I forgot the name.
Isn't it
Talentola? No, Luticola talentola, because the surface remains to a gecko. The skin of a gecko is very, it's not smooth. So, and they are living in the same area, but for us it's very rare because it's in a cave around 2,000 meter up.
Yes.
In very strange. So we think it's probably a product of a period of very cold climate window that we have a long time ago in the Canary Islands.
Yes. So this is the same with many areas. We described 3 new species in Galapagos, and they've been living in a quite harsh environment. We've described in Madagascar new species, and especially in the lava tunnels, because it's shadowy. So this could also be some kind of challenge for and which seems like in Antarctica low temperature, in low light intensity in the tunnels, in caves, it makes suitable this for Luticola, which is very resistant to also to human impact. So it seems like a wild boar of of the diatoms, the most resistant species and very easy to adapt to any conditions facing, and like humans also. Adaptation capability is very high, right?
Yeah.
It's very interesting.
Yeah, it is. So I have the question of Nurul now here, of regarding to the toxic microalgae. Can you listen me? Can you hear me? Yeah.
Yes. Yes.
Nurul, okay. Nurul, well, your question is if
Yes,
is there any characteristic to identify or mark the toxic microalgae? Well, there are two ways just to produce or establish a culture and analyze the toxins produced by these cells, or the other way is the normal way, is observe the symptoms of the fauna and flora that they produce also in the human health. Normally when you feel sick, you think, oh, probably I eat something, or I was swimming in a special part, and the history starts from the end, not from the beginning. So you have two different ways. Analyzing the toxin from a culture, or observing the symptoms that they produce in the ecosystem and of course human health. And of course, not all the species in the same genus produce toxins. For instance, in Gambierdiscus, there are a few number of species that they produce ciguatoxins, or in Ostreopsis, there are a few species that they produce palytoxins. So also, and not ever they produce the same level or amount of toxins. Also, is depending of the environmental factors. When they are stressed, or when they are reproducing, they produce different levels of toxins. So that's why it's so important to monitoring and controlling the factors, and of course the population of these cells in the community. Because we are talking about a cost annually in the world is around 4 billion with B billion of dollars in every year. So imagine how important is, in the point of view of economical point of view, to control this problem. I don't know if I answered your question.
Yeah, I get what you say, but I really want to know that is there any ways that, firstly, we can know, oh yeah, this is the toxic one, or yeah, no, this is not non-toxic, just like that. Is there any ways to know that easily for the first time?
Yeah, well, it's easy if you are a taxonomist or systematic, you are involved in this amazing world. It's easy when you are, you know the special characteristics and you have a very deep knowledge about the species and the genus. So that's why it's so important for the students and the people is now the new generation study the basic biology, study the systematic, because now it's a fashion about the molecular biology and everything, but we cannot forget the basic of the biology. It's, for instance, the taxonomy. If you have very clear what is the difference between the species and what is involved, talking about toxin of this species, for you will be very, very easy when you discover under the microscope oh, this is the special feature of this genus that produce toxins. So you have to be a fan of the taxonomy, and of course study. This is a very amazing field, and of course you have a lot of possibilities in your country, because it's like Dr. Witkowski was talking about. Your Indonesia is full of habitats, full of conditions. So I think it's very important monitoring and control this, because a lot of population is living very close to the coast. No? So don't forget the taxonomy, don't forget the systematics.
Yeah. So the only way is to study about it.
Yes, it's clear, I think.
Thank you so much, Mr. Emilio.
Sorry?
Pak Otias, can I raise questions?
Okay, short question, Bu Umi, as time is limited.
Yes, thank you. I have a question for Bu Yenny, Prof. Yenny, and Emilio. Prof. Yenny, as far as I acknowledge, shrimp got like non-specific immune response. So it's always like forgetting things, forgetting what is the disease or the that attack them, and they always like forgetting. They cannot remember. Now, suppose your technique you apply it for the the juveniles of the shrimps. Can they remember along their growth, even if they rare in different places? For instance, like you applied your technique in hatchery of something, and then this juvenile rare in some other pond, in some other area. Do you think that this shrimp will still remember the the increased immune system that you have applied for them? Thank you. And my question for Emilio is, I also interested in HABs, because I was once research on HABs in Ambon Bay. And it seems like from the research, the HABs always happen periodically, you know, like once in a certain period of time, they always like happen again. And it's because of the the cysts that they sink on the bottom of the sediment, where they always like blooming. Is that happen also in Canary Island, or with, in the research locations of yours? Thank you.
Thank you for your question. No, well, our condition is different. We haven't rivers in Canary Islands. We haven't a lot of rain like in your country. So our conditions are related with especially winter temperatures. It's funny, but when we have a cold winter, we have normally a very, very extreme summer with a lot of blooms, because dust storms and, of course, high temperatures and high irradiation. Nothing related with sedimentation, because we haven't rivers here, and our system is very, very simple. It's oligotrophic. So the problem of in oligotrophic areas is when the changes are very, very small. You can observe very big variation after 2 or 3 days after these changes. So in Canary Islands, for us, the most critical period is the winter time. In the cold winter means a very relaxed year with a very high temperature, or temperate or warm winter means problems the rest of the year.
I thought the HABs.
So sorry, excuse me.
Oh, yes.
So this is very important. Extrapolation is normally dangerous in our field because your area, your islands have a different condition of others. So of course you have to read, you have to know all the possibilities, but never think about other places. Think about only your area and what is the important factors that they are producing this problem.
Oh, okay, thank you, Emilio. Do you think that they could bloom because of only the temperatures, not without nutrients?
Sorry, I can.
Do you think that they bloomed just because of the temperatures, not the nutrient as well?
No, not only one factor. Temperatures, of course, is most important or the most important, but of course the nutrients. Remember, they need nitrogen, phosphorus, and carbon. Of course, iron, but also they need a temperature to grow in the better condition, and of course the day length, the radiation, and of course, and that also is important, the relationship within the community, because we don't know it, but there is a big war in the sea. They are fighting each other to get the better place. So temperature is very important. Light is very important. Wave exposition is very important. Nutrients, depending of the genus, also is very important. For instance, for Gambier discus, it's not so interesting, the nutrients, because it's a very soft and very quiet genus. But for instance, for Ostreopsis, for other planktonic species, nutrients are really, really interesting. So again, you have to focus your research in the genus. Not every genus has the same behavior. Not every genus needs the same one. So that was my presentation. Ostreopsis needs temperature, light, nutrients, and wave exposition, and also a life cycle. Life cycle is also interesting. But for instance, Gambier discus only needs a quiet places with a temperature very, very, ¿cómo se dice?
Low,
No,
mild.
Depending. They prefer low or high, but in Gambier discus, temperature is important. In Ostreopsis, it's a mix of factors. So you have to know the necessity of each one.
Okay.
Okay.
Thank you.
Thank you, Bumi. And now, sorry, Bumi, because it's we already 3 hours from 2 and 5 here. So I think you can ask privately to the Prof. Yenny about your question today. Okay, sorry, I'm sorry. So everyone, thank you for coming today. And before we close our seminar today, so I want you open your camera, and we will take a photo session, maybe a while, and Adrian will guide us to this session. Please, Adrian.
Okay. Please put the camera on. Ready?
Ready.
I'll do the count. 1, 2, 3.
Thank you.
Again. 1, 2, 3.
Okay. Shall we smile?
Yes, of course.
All of you are smile.
Yes.
Okay, Adrian, is enough?
Okay, once more.
Once more, okay.
Smile.
More smile.
1,
1, 2,
3.
3.
Thank you. Okay, yes.
Okay. Yeah.
One request to you.
Yes, Professor.
Can you send me, please, the high resolution of the background? My screen area. I will print it.
Okay.
And make a poster.
Okay. Okay. I know because this is Raja Ampat, right?
Yes. So I really would love to have this on in our lab and of course in my house.
Okay. Okay. Thank you. So we will close this session. So thanks you all speakers for interest share on this occasion, and thanks.
And we speak soon, right?
Yeah.
Yeah.
So
okay.
Thank you very much.
Thank you.
Thank you.
Yeah.
All.
Is Yunyanta still there?
Thank you.
Yunyanta.
May I close this session now? Okay, please. Okay. So thank you for our speaker, and thank you especially for Prof. Yenny for as a chairman, world class professor program today, and to all our participants this evening was very enthusiastic following the seminar and workshop to the end of the agendas. So I'm Octius. Please forgive me if any shortcoming from me. See you next week. See you next week.
Don't say this.
Yeah.
Apologies for my late arrival. Yes.
Okay, Professor. Thank you, everyone. Wassalamualaikum warahmatullahi wabarakatuh.
Assalamualaikum warahmatullahi wabarakatuh.
So it's free. You can talk each other.
Yenny.
Yes. Yes, Andre.
Please say, Yunyanta, that my ginger is growing very nicely.
Yeah, okay.
I wasn't aware that it grows also in the soil.
Yeah.
You put it on the. You grow it in the kitchen or?
In my kitchen, my window, and also curcuma. And yesterday I spotted that my ginger grows underneath
saya pamit dulu ya, Bu Yenny.
Oh ya, terima kasih Bu Umi.
You can try in the forest.
Terima kasih Bu Umi.
Ya, sama-sama.
Bu Umi next week ya.
Insya